Is that not what you wanted? That is what you wanted. Okay. Ruined the whole 3-day weekend, right? No? So the average on the exam was 62, which is just about what it's been on the last couple of exams. No significant changes. I was a little disappointed, I have to say, in performance. I expected a little higher performance, but I also recognize that it is the end of the term, and everything is piling up, so that happens. And plus, there's a 3-day weekend coming up, which we all have our minds on, I think, which is kind of odd if you think about it. You know, it's one day, one extra day that you get. It's not like you get 2 weeks or something like that, but you get one extra day. But somehow it just, in my mind at least, seems bigger than all of that. So anyway, hang around afterwards, and I will give you your exam. We are embarking today on talking about the citric acid cycle. And the citric acid cycle is, again, a cycle you probably get exposed to in a lot of different classes. So I'm going to go through it fairly quickly. I will not hold you responsible for structures on the citric acid cycle. Okay? Unless I change my mind. But I don't think I will. So, all right. Like glycolysis, the citric acid cycle is what we call a central metabolic pathway. And a central metabolic pathway is one that is called that because it's central to the metabolism of quite a few organisms. It's rare that you see an organism, extremely rare that you would see an organism that wouldn't have a complete citric acid cycle. The citric acid cycle is an example of a pathway that we would refer to as anaplerotic. A-N-A-P-L-E-R-O-T-I-C. Anaplerotic pathways are pathways that exchange materials with a lot of other pathways. So as we shall see, the citric acid cycle pathway, for example, provides intermediates for making amino acids. It's also a pathway where amino acids are broken down through. That is, they break down into intermediates in the citric acid cycle. Okay? So there are other things. The Succinyl Coa, for example, is used in the synthesis of heme, which is an important intermediate there. And citrate itself serves as an allosteric regulator of fatty acid metabolism. And we'll talk about that when we talk about fatty acid metabolism. So it's very central. Fatty acids themselves are broken down into acetyl-CoA, and acetyl-CoA is what we will think of as a starting point for the citric acid cycle. So these are all, the citric acid cycle is very intimately integrated into the cell. Very, very important. Okay. The citric acid cycle occurs in the mitochondrion. All of the enzymes for the mitochondrion are, for the citric acid cycle, are found in the liquid portion of the mitochondrion. We will refer to it here as the matrix, okay, which is really the innermost part of the mitochondrion. The one exception, yeah. The enzymes of the citric acid cycle are found in the matrix of the citric acid cycle. Okay? And matrix of the mitochondria. God, you can see I'm ready for this with this thing too. All right. Now, with one exception. And the one exception is that there's one enzyme that is found in the inner membrane of the mitochondrion, and that's succinyl dehydrogenase. Succinate dehydrogenase. There we go. Thank you. S-U-C-C-I-N-A-T-E. Dehydrogenase. D-E-H-Y-D-R-O-G-E-N-A-S-E. Okay? Now, like glycolysis, I think you should know the names of the intermediates, and I think you should know the names of the enzymes. Okay? And again, you're welcome to use any abbreviations that I use. There tend not to be too many abbreviations in this cycle, but I will use them as appropriate. Okay. Now, starting with the mitochondrion, here is the structure of the mitochondrion. And the mitochondrion has, for our purposes, some very significant elements that must be intact in order for the mitochondrion to function. The mitochondrion is described as the sort of fuel station of the cell. It's where the vast majority of the ATP is made. We'll talk about the electron transport system and oxidative phosphorylation next week. But it's in the mitochondrion where most of the ATP is made. And the synthesis of ATP really relies on this inner mitochondrial membrane. You can see it as sort of these little invaginations going in like this. And what these provide are additional surface area, which is very, very important for the electron transport system and for oxidative phosphorylation. We're not focused on that yet. We're focused on the metabolism, that is the citric acid cycle that's happening inside of this matrix. And as I said, the matrix is the sort of liquid that's in the middle of this whole gamish. Okay. We'll talk more about the anatomy of the mitochondrion later when we talk about the electron transport system. But I just want to give you a placement where all these things are occurring. Okay. The citric acid cycle from above looks like this. And it starts, at least in this scheme, with pyruvate. And the only reason we're showing it starting from pyruvate is because we've just finished making pyruvate in glycolysis. Okay? There are many, many ways for things to get into the citric acid cycle. So we're only looking at one of them here. And since it is a cyclic pathway, meaning it goes around and around and around, technically there isn't really a starting point or an ending point. But we will talk about the cycle as if it starts right here. Because acetyl-CoA is one of the most common intermediates found in cells. It's found in the breakdown of fatty acids. It's found in the breakdown of many amino acids. And it's also found in the breakdown of glucose to form pyruvate, and pyruvate itself gets broken down to acetyl-CoA. Okay. Let's start with that reaction first. So we finished glycolysis talking about how we had 2 molecules of pyruvate from 1 molecule of glucose. And depending upon whether or not the cell had oxygen, the cell could either do oxidative processes, which involve the citric acid cycle, or fermentative processes, which involve either lactate or ethanol, depending upon the organism that we're talking about. Okay? The pathway that we're talking about here will only occur, as we'll see later, if oxygen is abundant. Okay? Oxygen must be abundant, and we'll see very quickly why that's the case. Okay? Pyruvate, you may recall, is found in the cytoplasm, because that's where glycolysis is occurring. And that means that in order for this process to occur, that pyruvate must somehow get into the mitochondrion. Well, pyruvate actually does get into the mitochondrion. There's a transport system that allows it to move across the membrane. And that transport system gets pyruvate in, where it encounters an enzyme known as Pyruvate Dehydrogenase complex. The Pyruvate Dehydrogenase complex, and that's Pyruvate Dehydrogenase right there. Okay? Gesundheit. Pyruvate Dehydrogenase is an enzyme that catalyzes an oxidation of pyruvate. This is an oxidation of pyruvate. The oxidation results in the decarboxylation of pyruvate. That's the carbon dioxide you see going right there. Plus, NAD gets converted into NADH. Okay? So we have an oxidative decarboxylation occurring. And the product of that oxidative decarboxylation is acetyl-CoA. So the 2 carbons from pyruvate make up the acetate part of it, and coenzyme A gets joined in the process. Okay. Now, I'll come back and talk later in a little bit more depth about the pyruvate dehydrogenase complex. Its mechanism of catalysis is interesting and kind of complicated, and I'll try to keep it simple for you. But it is an enzyme that uses many coenzymes. It actually uses 5 coenzymes to catalyze the reaction that you see to make acetyl-CoA. Well, just stepping you through the citric acid cycle, as I said, I'll come back and talk about pyruvate dehydrogenase. To step you through the cycle, I want you to focus up here. And notice that the acetyl-CoA has to enter the cycle at some point. Acetyl-CoA enters the cycle by being joined to a 4-carbon molecule called oxaloacetate. And you've seen oxaloacetate before. What pathway had oxaloacetate? Gluconeogenesis, right? Okay. So gluconeogenesis had oxaloacetate in it. And you may recall that the pyruvate carboxylase enzyme was in the mitochondrion. So this was a source of oxaloacetate. All right. So there's oxaloacetate, a 4-carbon molecule, combines with a 2-carbon molecule. And that results in a 6-carbon molecule and the splitting out of coenzyme A. And that's labeled there as COASH. That's a way that's sometimes abbreviated. Okay. The 6-carbon molecule is known as citrate. It's what gives the pathway its name. Today we call the pathway the citric acid cycle. It's been called by many names. It used to be called the Krebs cycle. Okay. It used to be called the TCA cycle, but today we pretty much refer to it as the citric acid cycle. Okay? Well, at this point we have citric acid in the form of citrate, and though I don't require you to memorize the structures, I think you can look at these structures and say, wow, it's not overly complicated if I had to memorize it. Look at this. Carbon, 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 carboxyl, 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 and the middle one has a hydroxyl. Right? I think you should know how many carbons are in each of the molecules here, because we'll see that changes as we go through. So you should know how many carbons are found in each molecule. Citrate, of course, has 6 carbons. In the next reaction, we simply see a rearrangement of the citrate to form something called isocitrate, which simply means we're moving the hydroxyl group. I'll talk about the enzymes later. Then we get another oxidative decarboxylation. So we're losing one of the carbons. There's carbon dioxide right there. That leaves behind a 5-carbon molecule. We have another oxidative decarboxylation, which releases another carbon dioxide and leaves us with a 4-carbon molecule, in this case attached to coenzyme A again. And then we generate a GTP. Okay? We're generating a GTP. This is one of the ways we can make GTP in the cell. Okay? And that also results in the release of coenzyme A. Leaves behind a 4-carbon molecule. At this point, everything else that's involved in the pathway is simply getting back to oxaloacetate. So we do one oxidation, we do a second oxidation, and we're back at oxaloacetate. Okay? That's the pathway in summary. So we started with a 4-carbon molecule. We added 2 carbons to it. We lost 2 carbon dioxides, and we ended up with a 4-carbon molecule again. Students sometimes get confused because they say, when they discover, for example, that the 2 carbons that you put in are not the 2 carbons that you lose. The 2 carbons that you added don't start getting lost until the second time around the cycle. Okay. Now, let's talk about that pyruvate dehydrogenase complex, and I'll give you a little bit of a heads up on confusion that arises with this sometimes. The complex is a complex of several things, and that complex, one of the things, has an activity associated with it called pyruvate decarboxylase. Okay? I tend to avoid using that term unless I'm simply talking about the reaction of the loss of the carbon dioxide. For the most part, I will refer to it as the pyruvate dehydrogenase complex, but some books will talk about pyruvate decarboxylase, and then they get the names confused, and they get the students confused as well. We don't want to do that. So the pyruvate dehydrogenase complex is what catalyzes the oxidative decarboxylation of pyruvate to form acetyl-CoA. Now, as I said, this enzyme uses multiple subunits to accomplish what it does, and you can see the subunits actually listed up here. Okay? They include a molecule called thiamine pyrophosphate, which sounds like thiamine, which is one of the reasons you need thiamine in your diet. It's abbreviated as TPP. It requires the electron carrier FAD. It requires a molecule called lipoic acid, which you may have heard about before. It requires the electron carrier NAD. And last but not least, it requires coenzyme A because we're making acetyl-CoA. So those are the 5 coenzymes that this enzyme needs to accomplish the oxidative decarboxylation of pyruvate. All right. Now, this shows a rather complicated mechanism, and I'm not going to take you through the entire mechanism. I'm going to tell you a couple parts so that you understand why some of those coenzymes are needed. Okay? So one of those coenzymes was thiamine pyrophosphate, and thiamine pyrophosphate is, of course, related to the vitamin known as thiamine. And thiamine pyrophosphate has a very simple function in this overall scheme. Okay? It turns out that the decarboxylation and oxidation of pyruvate occur in 2 separate steps. So I talk about it as if it's oxidative decarboxylation, as if it's one big thing, but it's actually 2 steps. The decarboxylation is the first step, and the oxidation is the second step. Okay? So if you have something that has 3 carbons and you decarboxylate it, you then have something that has 2 carbons. That 2-carbon intermediate is very reactive. And it's actually carried by thiamine pyrophosphate, which grabs a hold of it and keeps it from reacting. Okay? Thiamine pyrophosphate passes off that reactive 2-carbon piece. And by the way, don't worry about hydroxyethyl TPP. We're not going to talk about that. Okay? It hands off that 2-carbon piece to lipoic acid. So we see a sort of a passing this intermediate down the line. This enzyme has multiple components, and they have to move literally from one place to another. Okay? So, decarboxylation occurs. Thiamine pyrophosphate grabs the 2-carbon piece. The 2-carbon piece gets passed off to lipoic acid, and it's in the passing to lipoic acid that the oxidation occurs. So decarboxylation first, oxidation second. Okay? Once we have had the oxidation happen, all right, this means that the acetate part is done. It's been oxidized. That means something has been reduced. And what has been reduced is lipoic acid. Lipoic acid got reduced. You'll see it started over here as a disulfide, but when it grabbed a hold of that 2-carbon piece, it became a sulfhydryl, 2 sulfhydryls. That means reduction. It takes electrons to do that. And where do those electrons come from? They came from the 2-carbon piece. Okay? Once this has happened, the 2-carbon piece says, okay, you're no longer reactive. I'm going to pass you off to coenzyme A, and we've made our acetyl-CoA. Well, what about those electrons? Those electrons, remember we started with a disulfide bond, and right now we have 2 sulfhydryl bonds. Those 2 sulfhydryl bonds are anxious to get rid of their electrons, and so what do? They pass them off to electron carriers, FAD and NAD. Ultimately, the product is NADH. Okay? So we can see something in this pathway about the role of each thing. Thiamine pyrophosphate carrying the 2-carbon piece, passing it off to lipoic acid where the oxidation occurs. The oxidation occurs on lipoic acid. And then lipoic acid passes its electrons off to FAD and NAD, ultimately making NADH, and in the process, acetyl-CoA is released. Okay. Now, why do I waste time on that? Well, A, you see something about the involvement of vitamins and micronutrients in a very important metabolic pathway. And second, there's another enzyme that we're going to talk about in the citric acid cycle that uses the almost identical mechanism, and it has the identical coenzymes here. So if you learn this one, you learn that one as well. Well, let's talk about the reactions. I'll tell you just a little bit about the reactions going through. The reactions that I described before, I will now tell you about. The first reaction I talked about was the reaction where we made citrate. And the name of this enzyme is very simple, folks. It's called Citrate Synthase, meaning it synthesizes citrate. This reaction is energetically very favorable. Okay? And the reason it's energetically very favorable is because this bond between the sulfur and the two-carbon piece, which is missing a carbon, it would appear. I just noticed that. Between the sulfur and that two-carbon piece, that's a very high energy bond. And so it's going to use the energy of that bond to donate a part of itself to something else. Does anybody remember the term I gave to that last time I lectured? Something that has a high energy bond and uses the energy of that bond to donate a part of itself to something else. Anybody remember? It'd be awful to give somebody 2 points of extra credit for it. Is it what? It's not oxidation, no. What's that? Translocation? No. It's a name for something. That's not a process. It's a name for something. A molecule that has this property. Yes? Activated intermediate. 2 points. All right. See me after class, okay? Activated intermediate is a molecule that has that property. And acetyl-CoA is an activated intermediate. Okay? I don't know where that structure came from. COH3. I think there's supposed to be a CH3 there that somebody overlooked. But anyway, I'll fix that structure. Okay. That energy for this reaction is largely driven by the breaking of that bond that makes possible the formation of citrate and the release of coenzyme A. We'll see later another reaction where coenzyme A is releasing energy to actually make GTP. So that's a very high energy kind of bond. Okay. The next reaction is the only reaction in the citric acid cycle that has a name of an enzyme that doesn't tell you what the hell it does. Okay? This enzyme name is a conatase. And in a sense it does because it actually has an intermediate called a conatate, but you wouldn't know that since I'm not showing you the mechanism. Okay? A conatase catalyzes the rearrangement of that hydroxyl group on citrate to make isocitrate. Okay? And though there's much more I can tell you about that, I'll spare it for you because it's Friday. I'm not going to talk about fluorocitrate. Okay. The next reaction is the first of two reactions that are oxidative decarboxylations. The first of two reactions that are oxidative decarboxylations. Okay? This reaction is catalyzed by the enzyme known as Isocitrate Dehydrogenase. And a dehydrogenase will always catalyze an oxidation. It will always catalyze an oxidation. And you can see here's an oxidation going on because there's NAD accepting electrons of accepting electrons of isocitrate to make NADH. Don't worry about oxaloacetate. We're not going to talk about that. The product of this reaction is NADH. It's also carbon dioxide and alpha-ketoglutarate. Okay? At this point, we have a 5-carbon intermediate. We've lost one of the carbons of isocitrate. Okay? In the next reaction, we have another oxidative decarboxylation, and alpha-ketoglutarate gets decarboxylated to form Succinyl Coa. And if you look up here, you'll see a variety of coenzymes that look like the coenzymes of pyruvate dehydrogenase. This enzyme is known as alpha-ketoglutarate dehydrogenase. I've abbreviated it here as alpha-KGDH. And you can also abbreviate alpha-ketoglutarate as alpha-KG if you want. Okay? So we see oxidative decarboxylation going on. Well, this is the enzyme that has almost the identical mechanism as the pyruvate dehydrogenase did for its reaction. And of course, it has the same 5 coenzymes. 1, 2, 3, 4, 5. Okay. Now, notice that this is an oxidation, and oxidation reactions in general tend to release energy, and that turns out to be very important because it's necessary to input some energy to make this high energy bond with in Succinyl Coa, because the Coa bond is a high energy bond. Okay. We can see that the Coa bond to the succinate is high energy by virtue of the fact that when we release the Coa, which is what happens in this reaction, when we release the Coa, there's enough energy to make a GTP. The energy released by breaking that Coa bond allows for the synthesis of GTP. Now, you might look at this enzyme name and say, what the hey, right? The name of this enzyme is Succinyl Coa synthetase. What do synthetases do? They synthesize something. What are we doing here? Are we making Succinyl Coa? We're breaking it down. So why do these idiots name it Succinyl Coa synthetase? Well, remember that enzymatic reactions are reversible. Gesundheit. Enzymatic reactions are reversible. And when they first discovered the enzyme, they were studying the reverse reaction. That's why it's called Succinyl Coa synthetase, even though in the direction that we're studying it, it's going forwards to make succinate. Okay, well, as I noted on the cycle earlier, succinate's a 4-carbon molecule. The rest of the reactions now are involved in getting us back to oxaloacetate. This is going to require 2 oxidations and addition of a water. The first of these oxidations occurs right here. It's catalyzed by the enzyme Succinate Dehydrogenase. And by the way, wherever I use the term dehydrogenase, you can always write DH. So if you want to call this Succinate DH, that would be okay. It's a designated hitter in baseball, but that's a different matter. Okay. This is the only enzyme of the citric acid cycle that's not found in the matrix. This one is embedded in the inner membrane of the mitochondrion. It's embedded in the wall, the inner membrane of the mitochondrion. And this is an interesting reaction in that it uses FAD as the electron carrier. Okay? We'll see that there's a parallel for this when we talk about fatty acid oxidation later, but this FAD being an electron carrier is there for a purpose, the purpose being that oxidizing these 2 carbons is not an easy task to do, and it takes an FAD to actually grab those electrons. We see that happening here, and evidence for that by virtue of the fact that we have a double bond that's been formed, and the hydrogens have been pulled off and put onto FAD in addition to the electrons that were in that bond to begin with. At this point, we have a 4-carbon molecule called fumarate. And in the next step, you may remember from your organic chemistry, I hope, that double bonds are very susceptible to addition of water to them. And so we see addition of water. By the way, notice this is a trans bond. For our purposes right now, it's not very important. But when we talk about fatty acid oxidation later, that trans bond will be important. Okay? Nonetheless, we have a double bond. It's chemically reactive with water. We can add water across that double bond where hydrogen will go on one of the carbons of the double bond, and the OH will go on the other carbon of the double bond. And this is catalyzed by an enzyme known as fumarase. Well, fumarase is acting on fumarate, so that's not totally surprising. And at this point, we have a molecule called L-malate. It's L because we have a carbon that has 4 different things on it. Okay? And we have an OH that will be good for us to oxidize. Now, the next reaction is, I think, one of the oddest reactions of the citric acid cycle. And I'll tell you why in a second. But this reaction converts L-malate into, oxaloacetate. It's catalyzed by an enzyme known as malate dehydrogenase. Malate dehydrogenase, or malate DH. In this reaction, we're seeing the alcohol on position 2 being oxidized to a ketone. We see the electrons being donated to NAD to make NADH. So that doesn't seem very odd. What's odd about this? Well, I'll tell you what's odd about it. This is a rare oxidation that is energetically not very favorable. The delta G0 prime for this reaction is positive, meaning that if we started with equal amounts of product and reactant, that the reaction would actually go backwards. Well, in the cell, we don't have equal amounts. And the reaction, as we will see in a minute, gets pulled forwards. It gets pulled forwards. Okay. That's the last thing I want to say when I show you the being pulled in just a second, and then we'll come back and talk about control. So the overall pathway was right here. We started here, we went all the way over here, we went all the way back over here, and now I've just described the last reaction to you, malate dehydrogenase, right here, and I said that this was energetically unfavorable. Okay? Well, how does the cycle keep going? The way that the cycle keeps going is actually with the next reaction. The cycle keeps going with the next reaction because the next reaction is very energetically favorable. It's a very energetically favorable reaction because, again, what are we doing? We're breaking a CoA bond. And that means that if this reaction number 1 is favorable, we're going to have very little oxaloacetate left in the cell. We're actually pulling this reaction, the malate dehydrogenase reaction, by removing the product. We're removing the product with an energetically favorable reaction with the citrate synthase. And because of that, the overall cycle proceeds along its merry way quite readily. Now, I told you that the citric acid cycle generated a lot of ATP, but so far you haven't even seen an ATP. You've seen one GTP, and you might can tell you, well, that actually expands just a little bit because if we think about GTP, we think about what we start with, it takes 2 pyruvates from 1 glucose. So for 1 glucose, it means we've got to have 2 rounds of the cycle, which means we're basically going to have 2 pyruvates, 2 GTPs that are produced. But still, where's all the ATP? The ATP is not made directly in the cycle. The ATP is made when the electrons from NADH and FADH2 are donated to the electron transport system. That's where the energy of the pathway is actually realized. Well, let's tally up what we've made so far, all right? Starting at pyruvate, or actually, let's say starting with glucose. We start with glucose, we had 2 pyruvates, right? 2 pyruvates are going to generate 2 acetyl-CoAs, and that's going to generate 2 NADHs. There are 3 reactions in the pathway that generate NADH. So if we multiply those times 2, we generate 6 per glucose. Add that to the 2 that we had up here. We now have 8 NADHs generated in the pathway. We also generated an FADH2, and since we go twice, that means we get 2 FADH2s. And of course, we have to remember the 2 GTPs that we made, which are equivalent to ATP. if we do the math, and we use some assumptions about this, and people argue about the assumptions, which is why the numbers of ATPs per glucose that you see reported vary. But if we make a reasonable assumption about the ATPs generated by the release of electrons of NADH and FADH2, what we discover is that starting with glucose, as I mentioned the other day, and going all the way to carbon dioxide, generates a total of 38 ATPs per glucose when oxygen is present. And oxygen has to be present in order for this pathway to occur. That contrasts with the 2 ATPs that you saw in glycolysis if oxygen was absent. So a lot more ATP generated if oxygen is present. Okay. Okay. All right. I'll stop and ask if you have any questions. Or do you want to just get the hell out of here? I think it's the latter. Okay. All right. Let me see. I'll tell you what I'll do. I will talk very briefly about the control of the cycle. I will very briefly talk about the role in anabolism, and I'll save glyoxylate cycle for Tuesday. How's that? Okay? But you have to sing loudly on the song. What's that? Okay. All right. Control the cycle. You've seen that glycolysis was controlled by controlling 3 different enzymes, right? And you might think, oh boy, the citric acid cycle is going to be a pain. It turns out the citric acid cycle's regulation is a lot simpler to learn than what's actually up here. Okay? It's a lot simpler to learn. It turns out that the citric acid cycle is largely controlled by the amount of NAD and FAD that's available. It's largely controlled by the amount of NAD and FAD that's available. How do we know how much is available? Well, in general, the oxygen supply to the cell will determine how much NAD and FAD is available. If there's plenty of oxygen, there's plenty of NAD and FAD, and that means that the citric acid cycle will be going. And if there's not enough oxygen, then there will be more NADH and more FADH2, and there won't be any NAD for those oxidative reactions, and the cycle will slow down or stop. So I don't care that you memorize which enzymes are stimulated, which enzymes are inactivated. I think you should know that any reaction that requires NAD or FAD will be regulated because it's regulated by what I just told you, the lack of NAD or FAD. Okay? But other than that, we're not going to worry about ATP, ADP, NADH, blah, blah, blah. We're talking about the availability of NAD or FAD. Let's keep it simple. Okay. There's the energy of the pathway. And here's that malate reaction I was telling you. That's the delta G0 prime of that reaction. Look at how positive that is. For an oxidative reaction, it's a very unusual oxidative reaction that's occurring. But the reaction that occurs after it is this guy right here, which is very negative, which overcomes it. And that's how the pathway essentially keeps going. Okay. The last thing I want to do is talk a little bit about what we call anabolism. And, of course, you know anabolism is the building up of bigger molecules from smaller molecules. And you can see that, I should also say this is, this is actually showing anabolism here. So we can see, for example, this thing called transamination off to the right. We'll talk about transamination when I talk about nitrogen metabolism, but transamination is basically the way that we make amino acids. It turns out that alpha-ketoglutarate, if we pull that carbonyl oxygen off and we replace it with an amine, we just make glutamate. I'll tell you about that later, okay? I'm just telling you for your free information now. If I take oxaloacetate and I pull its carbonyl oxygen off and I replace it with an amine, I just made aspartic acid. If I take pyruvate which isn't even part of the cycle. But if I take pyruvate and I pull its carbonyl oxygen off and replace it with an amine, I make alanine. So we start seeing relationships between amino acids and several of the intermediates that are in this cycle. When we later talk about nucleotide metabolism, we'll see that fumarate plays a role in nucleotide metabolism. As I've already mentioned, Succinyl Coa plays a role in the synthesis of heme. Okay? So these intermediates are tied to many other pathways. And this is really important because we tend to think of pathways as very isolated and going on only within this pathway. But in fact, all these other reactions off to the side and so forth are also all occurring. So if the cell is needing to make glutamate, it can start sucking off this alpha-ketoglutarate and do just that. If the cell has too much glutamate, it can feed it back and reverse that reaction so that it starts making alpha-ketoglutarate, which ends up getting oxidized inside of this cycle. So this pathway is a good exchanger. Okay? It's a good exchanger. It's like a thrift shop. You go to a thrift shop to pick up something that somebody else has been using that still has some good use in it, right? And you take it home and use it. And then when you're done with it, what do? You take it back to the thrift shop, right? Well, that's what the citric acid cycle really is. It's a cellular thrift shop for exchanging things. Okay. I'll talk more about what's up here on the upper left in the lecture on Tuesday because that actually relates to the glyoxylate cycle. But you've got to hear the song. It's a cute song today. Okay. All right. Okay. This one's called the citric acid cycle. What did I just do? Sorry about that. Oh, what did I just do? There we go. It's a source of energy. It gets electrons moving while reducing NAD. It starts with citric acid, turning to a connotate, which becomes an isocitrate. Away glutarate. You notice I'm not singing at this point. All right, guys. See you on Tuesday. Oh, you want your test back? Oh, no wonder you're still hanging around here. Okay. Okay, I think it's just me today, so I'll have to